In this video, we will show you the power under the hood of K-Pro and the impact it can create for biopharma workflows. The first, very important pillar is the data layer. K-Pro can access huge amounts of multimodal patient data, both public but also proprietary from all the Owkin data network. Thousands of patients whose data can be licensed and connected to the product. Now let's test with an example on TROP2 expression across different cancers. K-Pro reasons through your prompt and calls for the proper data and tools to orchestrate the answer. You can look at its thought process and then look at the results. The most striking part is the ability to directly look at patient data and access insights with detailed clinical anchoring and population summary to best understand the scope of the insights that have been delivered. These insights are derived from bulk, single-cell, and even spatial transcriptomics. Grounded in data, you have the reasoning and interpretation of those results to help you move forward. Here is a more complex example that would take days without K-Pro. We ask for spatial expression. The tool can look at spatial transcriptomics, intermix aggregate data for tumor islet, stroma, and provide you a very detailed answer and reasoning about gene expression, heterogeneity or uniformity, and associated recommendations. To go even deeper, you can look at individual patient data to really understand. What does this mean? What's the exact H&E slide or spatial transcriptomics? How can this inform your thinking? And of course, all these results are always cross-referenced with the literature. Now, what if we want to productize this ADC thinking and write a complex recipe that can be repeated over time? That's what we call skills. Here is an example. We want to find the optimal subpopulation for a given ADC. This recipe will then start with the expected inputs and outputs to make the recipe work. Then we can define the steps of the workflow. What are the desired features? How do we extract them? What is the unsupervised subtyping I would like to do with those features? And so on. And for each of these steps, we can define in detail, with additional subskills, what the exact methods are that need to be used. We can then invoke the skill on one specific example. Here, TROP2 in triple-negative breast cancer. Fast-forwarded, all these steps will be executed one after another to provide all the results in one go. The thought process reminds you what has been done according to the recipe, and then you can look at the results. We can, for instance, investigate the different features that have been computed on all the patients in a heat map. Or we can also look at the different clusters that have been derived and the kind of ADC response that has been inferred by the machine learning algorithm. As for the conversational mode that we've seen before, on top of the plots, on top of the data, you'll of course access all the reasoning and the summary that helps you derive insights from the data, and the machine learning models that have been embedded in this recipe. Now, what if you want to scale this? You can use the campaign mode in order to use one given workflow, such as the ADC subtyping we discussed before, but in this case, you can scale that one in order to really multiply the number of research angles you can cover. Let's try, for instance, to generate 4 different IDs, and then for each of those IDs, we will be able to launch a dedicated effort. It is as powerful as having several K-Pros in parallel, each one running an independent sandbox. For each sandbox, you can access all the results, like a fully fledged research project. From intermediate results to final outputs, everything is there to inform your decision-making. And you can deep dive into those insights in the interactive mode that we've seen at the very beginning, from conversations to workflows to campaigns. This is how you can leverage K-Pro's powers.